Good morning, everyone. My name is Yasmin Rahimi. I'm a senior research analyst here at Roth Capital Partners. I cover biotech companies with special interest in orphan, ultra-orphan diseases. It's really a pleasure to have with us Frederic Charreau, President and CEO of Logic Biotherapeutics. So the format of this discussion is fireside chat, so feel free to interrupt me with questions or we can wait at the end. So Frederic, it's a pleasure to have you here. What is your lead asset, its mechanism of action, what does it treat, and where are you in clinical development?
Good morning, Jasmine. Thank you very much for inviting us today. So our lead asset is called LB001. It treats a disease called methylmalonic acidemia, or MMA, which is a rare or ultra-rare organic acidemia. And the mechanism of action is genome editing, and we are currently in a preclinical stage.
And before we kind of get into the technology, what are sort of key events or upcoming catalysts on your radar for the next 12 months?
So as a preclinical stage company, I would say the main catalyst for this year will be the filing of the IND by the end of this year for this disease, hopefully moving to the clinic soon after. In between now and then, obviously, there are a lot of things which are happening on the non-clinical side as well as on the manufacturing side. In parallel, we are expanding the platform. As I will tell you in a minute, our technology is very much a platform, and we have already demonstrated proof of concept in multiple animal disease models. We intend to continue that, take advantage of the learning, and declare our second candidate by the end of this year. And maybe the last, and hopefully milestones would be potentially a strategic partnership.
Thank you. So maybe can we spend some time, can you enlighten us on Logic's proprietary gene editing called GeneRight? Can you describe it, how it functions, how does it set apart from your competitors?
Sure. So we call it GeneRight because you take a ride on the gene, as you will see. Maybe I can step back a little bit and tell you where we fit in this genetic medicine world. I think many of you are aware of what we call canonical gene therapy or episomal expression gene therapy, most of the company you can name. And basically what they are doing, in a great way, is to deliver a transgene within the cells for the cells to be able to produce a missing protein or faulty proteins. It's great, but it does not answer every question, especially when the cells are dividing, which happens, in multiple organs in the liver and in the body, such as the liver, and especially in children, when the children are growing. Another potential pitfall of this canonical gene therapy is the fact that we will see, and we see in preclinical studies, some off-target integration of these AAVs. And one of the downsides of this off-targeting is the fact that if you carry a promoter with you next to an oncogenic site, you could trigger carcinogenicity, and it has been described in literature many, many times. So if you want to defeat this lack of durability when the cells are dividing or when the tissues are turning over, you need to integrate into the genome. That's when the genome editing technologies are very interesting. The first one you may have heard about are the CRISPR, TALENT, or nucleases, zinc fingers. And they are very interesting technologies, very potent, but sometimes not always very, very specific. And I would say that these two ideas that the team at Stanford in the lab of Mark Kay, who is the head of human gene therapy at Stanford University, had in mind when they developed GeneRight. So GeneRight is AAV mediated. We used AAVs to go into the cells, but instead of carrying a promoter and a nuclease, so we are promoterless, nuclease-free, we have a special cassette which allows us to do gene integration in a very specific locus by harnessing a naturally occurring process called homologous recombination.
And so after integration of the transgene downstream of the albumin promoter using GeneRight, how correlated are the circulating albumin levels to expression of the exogenous protein? Is it 1-to-1?
Yeah. So maybe I can tell you. So first, we target the liver. In the liver, we target albumin. In other tissues, we will target other genes. The way it works is basically by homologous recombination, we are able to insert at the very end of the albumin locus in the liver this transgene coupled with a small peptide called 2A peptide. When the protein is when there is protein translation, the full albumin protein is expressed, tagged with this small 2A peptide, and the 2A peptide plays the role of ribosomal skipping, or it allows bicistronic expression. So what we see, the ribosome will continue its work and will express the missing protein, the transgene you want to express, with a 1-to-1 ratio. The big advantage of this technology, this bicistronic expression, is that when the protein is not released in the circulation, we cannot dose it in the blood, and when it stays intracellular, we can look at the albumin 2A and use that as a diagnostic tool to show or prove that we have integrated and released the right protein at the right place. And yes, it's a 1-to-1 ratio.
So then after deciding, the gene is inserted correctly, what type of additional optimizations needs to take place to make sure that you're achieving optimal delivery of the GeneRight vector?
So I would say that the same type of optimization that other genetic medicine technology have to go through. We could optimize the gene, but I think what is interesting with GeneRight is the modularity of the platform. Once we have decided to go after a gene, in the case of the liver, it's the albumin, when we have defined these homology arms which are flanking the transgene and the 2A on both sides, we don't have much to do to go to the next indication. We just have to swap out the transgene, change it by another transgene for a molecule the protein we want to express. And sometimes we have to optimize a little bit the homology arms, but that's something we can do very quickly. And that's the way we have been able to generate relatively fast with partners or internally a lot of proof of concept in multiple diseases.
So when you're looking from clinical indications to use your novel technology, what sort of selection criteria do you use? Do you want to be a liver company? Is your technology specific for sort of liver therapeutics? So as you're expanding out, what are you thinking, and are there certain capabilities that you may not be able to expand into?
So for now, we have, I think we have developed a lot of skills and know-how in how to target the liver. Does that mean we want to be a liver company? Not really. I mean, you can target with this type of technology, you can target the liver to express protein which are useful for the liver, but also to express protein which are not needed by the liver, such as in hemophilia. It's not a liver disease per se. We just use the liver as a factory to manufacture the drug, basically. So when we look outside, obviously the liver is very important for us. Most of the proof of concept today has been done targeting the liver. But we are not intending to stay in the liver. We have already done some work in other tissues. Some of it has been published, and we intend to expand to other tissues very quickly.
Maybe we can pivot a little bit into LB001, and it's being developed for MMA. Can you maybe highlight to our audience, the high unmet medical need, the sort of prevalence rate around the disease, and what are the current therapeutic options available for these patients right now?
None. But so MMA is an ultra-orphan organic acidemia. It's a terrible disease caused by the lack of a gene called MUT, M-U-T. It's on the newborn screening, so you can detect the disease very quickly. Nevertheless, most of the time the mother has the feeling that there is something going wrong with the kids even before the newborn screening data come back. So by lacking this, the expression of this gene, the children cannot metabolize proteins. So if you don't give them proteins, they don't grow. If you give them proteins, you trigger a terrible storm of metabolic decompensation, which leads ultimately to coma and death. There is so no treatment for these patients today. The only tool the physician can use are high-calorie, low-protein diets. It's basically formulas, so these people have to to eat formulas all their life. Very often it goes through a GI tract, and as I was saying, there is no real alternative. Then at some point the liver transplantation, and even the liver transplantation isn't ideal. It's, as we know, it's a good surrogate, but for a treatment, but it has a lot of side effects, and the life expectancy of this patient is in between 20 and 30 years old, and they don't have a normal life as well as their family.
And what is the prevalence rate in the U.S. and Europe?
It's one incidence is 1 in 50,000 births, which means 1,500 to 2,000 patients in the U.S. and about 6,000 patients worldwide.
Thank you. And then, for our audience, they may have also looked at Moderna, who's also developing a therapy for MMA with an IND filing that did in January of this year. So can you kind of highlight maybe the differences of LB001 and GeneRight compared to the context of Moderna's approaches?
Yeah. So first, and I've been in the rare disease industry for decades now, and I think it's always good news when there are more than one company getting interested in a disease. In the case of MMA, what is interesting is the two companies disclosed their interest for MMA a couple of months apart, so a little bit of surprise. Moderna might be a great company because they have a French CEO, so I think it's a very good way to start. But I think the big difference between what we do and what Moderna is doing is the modality of treatment. If we are right, if we can show in humans what we have demonstrated in animals, which is that with one vector, one injection, you can provide a protein expression for life, and some of our mice now are older than 15, 16 months old, which is very old in the life of a mouse. It would be the perfect treatment for patients. Now, Moderna's technology is more a technology you will have to re-administrate every week, every month, to be defined. But I think, even if the patient population is small, there will be different modalities for different patients. And the ability to go early that we are claiming, and very, very young in the patient before some non-reversible sequelae occur, which is one of the major issues of this disease, I think is very interesting for GeneRight, and we should provide in this lasting effect should provide a very good efficacy to this drug.
And so as we're thinking, one of the challenges with gene therapies, like looking at the safety, right, from the preclinical and its transferability. So can you tell us a little bit, the advantages from a safety perspective of GeneRight versus other gene delivery modalities that are currently being pursued?
Yeah, so all these technologies are multi-components in some ways, and one of the components is the capsid, the vehicle you use to go within the cells. And here in our case, we are using a capsid called LK03, which is already in the clinic with Spark. Actually, that is the capsid that Spark is using in their hemophilia A compound, which has been proven to be a very efficacious and very safe capsid to date. So I think here we have de-risked a little bit of a big part of our platform. Now, as I was saying before, because we are harnessing this naturally occurring process, just to integrate the gene at the end of locus, we think that we are not too much prone for off-target integration, which is one of the downside of gene editing and gene therapy in general, as you know. So obviously, I mean, you need to go to human, and you need to have extensive experience to declare that you have a safe product. But to date, we haven't seen any safety concerns in any animal studies we have done.
And then can you remind our investors in regards to the IND filing, the timing for MMA, when you plan to do that, and additional indications sort of, but probably all eyes will be on the IND filing of MMA, right? Is this a 2019 event, 2020, we could do?
So our plan is still to file an IND by the end of this year, so 2019, to be in the clinic early 2020. In this type of diseases, you can see data because it's open label. It's going to be a small trial. I think you can see data relatively quickly, so we will have an idea of the efficacy of the drugs, the dose, and so on. So that's the main catalyst to keep an eye on, I would say. But in parallel, as you may know, we have proof of concept in hemophilia B, in alpha-1 antitrypsin, Krüger-Najar, which has been published in very good journals, and it's very possible that one of these programs is taken forward by us or by a partner in the coming year. And for the in-house program, we intend, as I said before, to declare our second indication by the end of this year also.
And then in regards to the, IND filing with MMA and your plans, can you kind of give a little bit color in terms of what sort of a clinical, target engagement analysis we would be seeing? Obviously, all eyes will be on the safety profile, but what would be ways to measure, gene rights, efficacy, and key biomarkers that you would be looking at?
Yeah, so first, as you outlined, it's a safety trial. Obviously we will look at some efficacy biomarkers. Here we should relate to what has been shown in preclinical studies. So we have biomarkers such as the circulating metabolites, like methyl citrate, MMA levels. There are many metabolites we can dose. This additional albumin 2A biomarker is also a very interesting tool that we will look at, and having that allows us not to go for biopsies, which is something you don't want to do in kids. Then you have more, let's call that clinical endpoints, such as growth, such as protein tolerance. So that's the type of things we're going to look at. And maybe you were talking about target engagement. We haven't talked too much about the details today, but it's known from biology 101, I would say, that homologous recombination is a relatively modest process in terms of efficiency. We do integrate in about 0.1 to 1% of the cells, but because we are taking a ride on the albumin gene, which is one of the most expressed genes in the body, we can obtain with 1% of the cells physiological levels of the missing factors and protein. What is interesting in MMA, and maybe coming back to one of your questions on selection, is we have seen what we call selective advantage. The cells which are modified over time, when the liver is turning around, are given a better health than the others, and they take over in the liver the non-modified cells. And what we can see after a few months, and it has been published and will be published again at ASGCT in a few months, we've seen that 25% of the liver has been modified, which is great news, and very it's the opposite of what we see in canonical gene therapy, where most of the time you can see the durability going down because being reduced because the protein expression is going down. What we have shown is, even if we start with a modest, nevertheless efficacious level of integration, and then a protein expression, it goes up, and it gives the liver, an even better advantage.
Thank you, Fred. I'm going to open up the session for questions for Fred. Paul?
So here in the liver, taking advantage of the output, moving the whole tissue, how hard is it to find like a suitable subscorer that has kind of beneficial level of expression?
Yes. Oh, sorry. The liver is the obvious tissue when you do gene therapy, because that's where everything goes, and also in our case, albumin is very, very expressed. And also, what I didn't mention, but which is very interesting too, it's very, very tissue specific. Albumin is not produced, or almost not produced outside of the liver. So when we look at other tissues, such as muscle, CNS, and others we have started to look at, we look for the equivalent of albumin, and I think that's your question. We are not disclosing which genes we are looking at, but I can tell you that in every tissue and every cell type, you can always find a gene which is overexpressed versus the other one. I mean, in the muscle, creatine kinase is the obvious one, but that's not the only one. So currently we're about to start, which type of modulation in terms of protein expression we could get by targeting such genes versus another one. But there are many other genes.
Thank you so much. So we'll take the time to thank Fred for being here, and thank you.